thank you, Mega Team, for inviting NCNED and for me to present some of our findings. And today in this presentation, I'll be talking more on NCNED findings on ME and long COVID findings using multimodal MRI. So I think Brianna, Stanley, and Etty had made my job easier because now I don't have to explain anything about the ME and long COVID. So, but what I would like to focus here is ME is a multi-body systems, and one of the most important symptoms experienced by ME is cognitive impairment. So, which is sometimes called the brain fog, and one of the main regions, one of the main body regions that is involved in the cognitive impairment is brain. And as we all know that there are more than 24 million people suffering from ME, and there is no clinical testing or evidence-based treatment, and all those people are diagnosed based on the international consensus criteria and Canadian consensus criteria. So after ME now there is a long COVID, and as Bray has already defined the definition of long COVID, I won't go that in detail, but there are more than 775 million worldwide suffering from COVID-19, and at least 10% of COVID-19 people are infected or maybe develop into long COVID in the future. And among the all common symptoms, as mentioned by Brianna, one of the main common symptoms between ME and long COVID is cognitive impairment. So if we talk about the cognitive dysfunction in ME and long COVID, again, as mentioned earlier, brain is the key body part that is involved in the cognitive impairment. So why do we need to study ME and long COVID together now? Because 13% of long COVID patients meet ME criteria. And as Brianna mentioned that they exhibit similar symptoms, as AT showed that there are similar TRPM3 dysfunction in both ME and long COVID. And 70% of ME patients report post-infectious onset. And if you look at the figure in the right-hand side over here, ME and long COVID, they have executive dysfunction, attention dysfunction, they have memory problems, as well as they have information processing speed. Information processing speed is very slow, both in ME and long COVID. So what's the motivation of our study is to study brain dysfunction in ME and long COVID condition. The goal or the aim of our study is to identify brain regions that are affected by ME and long COVID condition. How are we gonna do that? So we are using 7 Tesla MRI. So MRI is, I would like to say, it's a non-radiation technique. So there is no radiation involved when we are acquiring MRI data. But we are moving to the, if we know like there are different field strengths of MRI, one is 3 Tesla MRI, which is pretty much used clinically all over the world, and there is 7 Tesla MRI. Here we are using 7 Tesla MRI because it provides better quality images. Sometimes some of the structural alterations goes undetected in 3 Tesla MRI, but that can be captured in 7 Tesla MRI. And it shows the clear picture of different structural images. As you can see here, this is in the left hand side, this is 3 Tesla MRI data, and this is the 7 Tesla MRI data. And if you can see here, the white matter and gray matter are pretty much clear when we use the 7 Tesla MRI. And if you look at another one, which we call the functional MRI, that is used to measure the blood oxygen level dependent signal, as well as like which regions are connected or which regions are weaker or which regions are strongly connected. We use functional MRI technique, and you can see that again the data on the 70 and the 30, the data over 70 is pretty much of higher quality than the 3 Tesla MRI. And we use this technique to measure the connectivity between the different brain regions. So we can see here like which regions are connected, which regions are strongly connected, which regions are weakly connected. And this will depend on the task, that depends on the cognitive task that participant performs inside the scanner. So as we said, as I mentioned that previously, that the ME and long COVID experience the cognitive impairment. Then how can we measure that inside the scanner? Is that we perform some color task, which is called a strobe task. We give participant a bit challenging task, and the task is something like this. They can see inside the scanner, does the color of the upper or corresponds with the meaning of the lower, or the task will start automatically soon. It will be displayed in the scanner, and you can see the different stimuli inside the scanner. If you look at the C, this part is pretty much easier. And if you look at the N here, neutral, this is pretty much easier. You have to click the button yes or no. But if you look at this one that I have highlighted, it says the blue it reads as blue, and it's the color here, it says the green. So the people are easily confused here. It's pretty much cognitively challenged to get it right, because people just read the words blue and they click no, but we have to look at the color here, which is green, and the word says green. So it's a bit cognitively challenged. So we try to track these, or when people perform this task, we try to see which brain regions are connected. Or they are connected strongly, or they are connected weakly. So to test this task inside the scanner, one of our PhD students acquired data from 28 ME and 15 healthy control, and we do some image processing technique, and we see like which regions are connected, or weakly connected, or strongly connected. Then we found that over here, we found that brainstem and cerebrum regions in ME-CFS were significantly weakly. I would say they found very weaker connectivity between brainstem and cerebrum regions in ME. And we went further and we found, okay, what about if not the regions, look at the small voxels, or for understanding, I would say the pixels or the voxels. We again found that multiple brain regions were weakly connected in ME compared to the healthy control. And however, we also observed that there was some stronger connectivity between ME in different brain regions. And if you look at these regions, these are pretty much involved in the abnormal blood pressure, autonomic function, sleep quality, as well and cortical arousal levels affecting memory, learning, and problem solving. We see that there is some kind of connectivity deficit in the brain regions in ME when we compare to the healthy controls. Then we also perform some correlation analysis to establish the association between this functional connectivity and the duration of illness. And we found that when we have the duration of illness, we found that there were some positive correlations between insula and anterior cingulate cortex. And we also found that there was also some connectivity, weaker, or I would say the negative or the positive connectivity between the cerebrum regions to some of the cortical regions. So we also tested other connectivity with the impaired memory, and we found that again there was some connectivity deficits between the cerebellum regions, and we also found that there were also some negative correlations within the cerebellum region. This shows that within the cerebellum regions, when they have the impaired memory, there is the stronger and the negative, I would say the positive or the negative connectivity deficits in ME. So recently, the Baden, we published that this paper altered brain connectivity in long COVID during cognitive exertions. And here again, we show that in long COVID patient, we found that there is some connectivity deficits within the brainstem regions, that is from midbrain to the medulla. So this shows that there is something happening within the ME patient as well as in the long COVID patient, and we have shown that there is some kind of connectivity deficits within in the ME. So we have published, NCNED has published a lot with MRI findings in ME. Here these are our publication here, where we use the multimodal MRI to see structural signal intensity, different microstructural changes in the brain regions of ME. So But so far what we have shown is yes, there is some changes in the ME and as well as in the long COVID. Now, how about, do they have the similar changes or the different changes when we compare with the long COVID and ME CFS patients? So for this, we tested one region, which is known as the brainstem, because if we look at some of those brain regions findings from the ME and long COVID, we see significant deficit in the ME population. So here we choose the brainstem because brainstem is involved in multiple functions. For example, motor function, sleep-wake cycle, cardiorespiratory function, cognitive function, and these are all the common symptoms experienced by ME and long COVID people. And furthermore, there have been reported that SARS-CoV virus was present in the brainstem, as well as they found the pronounced inflammation in the pons, brainstem, and cerebellum. That's why we choose this brainstem as a test case. So we acquired the data. This is the pilot study. We acquired structural data from 7 Tesla MRI. We tested that in the 10 ME, 8 long COVID, and 10 healthy control. And these are all the parameters that we used to acquire the MRI data from 7 Tesla MRI. So here we compare the brainstem volumes in ME, long COVID, and healthy control. So the red one is ME, the green one is long COVID, and blue one is healthy controls. And here, if we look at the pons volume here, we found that there is significant difference between the ME and healthy control, long COVID and healthy control. However, there is no significant difference between ME and long COVID. Similarly, if we look at the whole brainstem volume, we found that similar results. We showed that there is significant difference between ME and healthy controls, long COVID and healthy controls, but not any significant difference between ME and long COVID. So we looked at the another regions, which is called superior cerebral peduncle, which is one of the brain regions that connect brainstem and cerebellum, and we found that superior cerebral peduncle volume was significantly higher only in long COVID patient compared to the healthy controls. And we studied the another region. We choose the another region is the hippocampus, because hippocampus is one of the main brain regions that is involved in the cognitive function as well as in the memory function. So that's why we say, okay, if both of them, if both ME and long COVID are having cognitive impairment, that could be due to the some structural changes in the hippocampus. So we use the same data, but here we increase the number. So we acquire data from 30 ME, 15 long COVID, and 15 healthy control. We use some fancy image processing technique to estimate all those soft fields volume, and we found that left cerviculum head in ME, which is in red again, long COVID in green, and healthy control in blue, showed that left cerviculum head volume was significantly larger in ME and long COVID when compared with healthy control. Similarly, if you look at another pre-cerviculum head volume, which was significantly larger again in ME and long COVID when we compared with healthy control. We looked at the further regions. Again, we found that left molecular hippocampal head volume and whole hippocampal head volume in left and right was significantly higher in ME and long COVID when compared with healthy controls. Interestingly, we did not find any significant difference in the hippocampal soft fields volume between long COVID and ME, and this shows that there is significant overlap or link between the ME and long COVID condition. If you look at the 80s results, also the TRPM3 channel was similarly dysfunction in ME and long COVID. So we also did some association correlation analysis between volume and the severity measures like pain in ME, and we found that as the severity increases, as the pain score increases, which is from 0 is the highest and 100 is the no pain. So we found that as the severity of the pain increases, pain level is higher, the volume goes down. Similarly, with the left pre-cerviculum head volume and with the left femur volume was also associated with the pain. We saw that as the pain level increases, your left femoral volume is also lower. So, but this was pretty much opposite in long COVID. We found that as the severity of pain increases, your volume also increases in left femora as well as in the right femora. So this is pretty much opposite. The pain association with the hippocampus volume is relationship is opposite in long COVID when we compare with the healthy control. So in addition to the 7 Tesla MRI study, we recently published another study looking at the brain neurochemicals in long COVID and ME/CFS. So here we acquired this data from 17 ME, 17 long COVID, and 10 healthy control. Had the 3T MRI, and we found that the glutamate level in ME and long COVID was significantly higher when compared with healthy controls. However, we found that N-acetyl aspartate, which is also known as NAA, was significantly higher only in long COVID when we compared with healthy controls. So it's not only the functional MRI, it's not only the structural MRI, even the brain neurochemicals are similarly dysfunctional, they're very much similar in long COVID and ME condition. So this again shows that there is some kind of overlap between these two conditions. Then we performed some correlation with the neurochemicals in ME, and we found that the physical function, unrefreshing sleep, and pain score were significantly associated with the brain neurochemicals. And again, with the long COVID, also we found that duration, physical function, and cognitive impairment were also significantly associated with the long COVID. So overall, we showed that because of there is some association between the severity measures and the neurochemicals as well as structural changes in ME and long COVID, this could be the reason why we are saying that some kind of symptoms, for example, sleep-wake cycle, cognitive impairment, both are impaired in both conditions. So in conclusion, what we have shown is that there is some functional and structural alteration in both long COVID and ME. And this could be the reason why people are experiencing the multiple symptoms in both conditions. Similarities in brain structural alteration and neurochemicals between long COVID and ME provide further evidence for a significant link between two conditions. Association between severity measures and brain impairment in both long COVID and ME patients suggest brain is affected by these conditions. These findings may be helpful to better understand the cognitive impairment in both conditions. So I would like to thank all the team at the NCNED, funding bodies, our collaborators, and all the participants who have participated in our study. And as he mentioned that we are always looking for the volunteers and we are running the clinical trial, please contact NCNED, and thank you.